Sri Gurubhyo Namaha. Welcome to your online teaching academy, and I welcome you all for the biology class. This is class 12 biology. It's the last part of the lesson, principle of inheritance and variation. We are going to see different types of disorders, and we call it as a Mendelian disorder. In Mendelian disorder, we are going to categorize on the basis of the chromosome, whether the disease is towards, the disorder is towards the autosome or allosomes. So first, it is sex-linked recessive disorder. Another one, autosome-linked recessive disorder. Actually, in human chromosomes, we have 23 pairs of chromosomes. In 23 pairs of chromosomes, 22 pairs are autosomes. This is very important to mention, pairs. And one pair allosomes, which determines the sex, whether the individual is male or female. This is female and this XY is male. Okay, the other body characters, otherwise you call it as body chromosomes, and this allosomes otherwise called as a sex chromosomes, right? So in Mendelian disorder, two things we are going to discuss. It's important 3-mark question, and how they are going to ask the question, mostly like symptoms, and then which chromosome, the number of the chromosome affected, and in which place it is, okay, the 22nd or the 21st pair, like that, right? So we'll see the examples here, a hemophilia and color blindness. In autosomes, sickle cell anemia, phenylketonuria, and thalassemia. Actually, this one we discussed already in the beginning. Still more we'll see here. First, let us talk about hemophilia, otherwise called as bleeder's disease or royal disease. It's a sex-linked recessive disorder. It's a very, very important term. Please make a note. We are going to deal with this term only, whether it is a recessive or a dominant. When we say recessive, when this particular disorder will exist only in recessive condition. Okay, for example, if the person, okay, I'll take it as XX female and XY male. Okay, and this disorder hemophilia, H, let me take H. If I write like this XXH, she is a carrier, okay? And if I'm going to write XHY, this person affected male, okay? Affected male. Actually, we said it is a sex-linked. So which sex-linked it is? It is X-linked. Actually, when you say sex-linked, there are two. Let me write here. Sex-linked to X-linked or Y-linked. Y-linked, called as Olhandric genes. Actually, I would have discussed this in the previous videos. X-linked, we are going to deal here. Y-linked, it's not in our syllabus. We are going to deal only with X-linked. So it is X-linked. Male have 2 chromosomes, in that only one X chromosome. So in this X chromosome, if there is a defect, okay, if there is a defect, then the person will be affected. Same if you take it female. In this female, this is male and this is female. In female, if only one X chromosome is having this type of disorder, then the person will be a carrier. Okay, the person will not be affected. When it is like this you call it as homozygous condition. When it is in homozygous condition, the person will be affected. Okay. Defect. So what is the defect? Hemophilia, we know where it causes the effect, in the X chromosome. Okay, and only in recessive condition, you can see here, only in recessive condition it shows the effect in female, whereas male it shows in one condition itself, because it is a X linked, only one X chromosome is there, right? The defect is blood coagulation, the blood won't clot. I told you it's the X-linked inheritance. The defective alleles produce non-functional protein. which later form a non-functional cascade reaction. In 11th class, we read about the cascade reaction. It's nothing but blood clotting. Okay, for blood clotting, there are 13 factors. Okay, out of these 13 factors, if any factor is not functioning well, it leads into bleeding. Okay, that type of disorder only we are calling it as hemophilia. Right, so the effect is here in the factor. Females suffer this disease only in homozygous condition. I told you here in this condition only. Okay, so it is, you can correct this, it is XHXH. Queen Victoria was a carrier, that's why we call this as royal disease. And here is a chart, and this is Queen Victoria's pedigree chart, this one. Okay, and here is a chart which represent this hemophilia. When a person, this is normal male. Okay, normal male. When a normal male marries a carrier female, okay, she's a carrier because it is a homozygous condition. Okay, what are their resultant progenies? Suppose if you find this is a difficult task to do, you can use a checkerboard. Okay, instead of doing this flow chart, you can go for a checkerboard like this. Okay, so it's easy. Who proposed this one? Punnett. So we call it as Punnett square also. Use the checkerboard. Okay, write the same thing. Okay, and then complete it. So you will get the result. So how many percentage of normal and carrier? Carrier, so here 50% normal. In this 50%, 25% normal daughter, 25% normal son. We started with carrier, I'll continue now. How many percentage carrier? 25% carrier and no down, and that 25% should be the female. If your answer getting male, then it is wrong. You'd have done some wrong calculation. So always a female will be a carrier because the only female will have 2 X chromosome. Okay. And how many percentage for affected male or female? Only 25%, that too only male. Okay, from this you may get a 2-mark question. Okay, we'll move to the next one. Sickle cell anemia, we discussed this one already. Sickle cell, so the cell is going to transform the shape of sickle. You can see here, this is a disc shaped structure. So what's this? It's RBC, and this RBC transformed to a sickle shape. How this transformation takes place? What is the core in this transformation? Is nothing but point mutation. We discussed in the last lesson also, right? I mean the last topic. So here is your textbook image. This is, now let us see this nucleotide. This one we call it as triplet codons. When there is a change in the codon, okay, one particular point there is a change. So this is our normal hemoglobin and it is a sickle cell hemoglobin. Okay, sickle cell hemoglobin means RBC naturally. So in this, it is GAG, okay, just observe this, and here it is GTG. So what is the change? Instead of A, okay, there is a point mutation and it is changed into T. Okay, because of that, see, the message read by this mRNA was wrong. It should be GAG, but it is GUG here. And because of this wrong transcription, there is a wrong translation. Instead of producing glutamine, it produces valine amino acid. Okay, and because of this, the structure transformed into sickle cell. Normally, it has to be disc-shaped structure so that it can receive the oxygen molecule. Imagine if it's a disc-shaped structure, they receive the oxygen molecules. We call this as oxyhemoglobin. You'd have read this in 10th standard also. Like this, like a plate. In a plate, only the food served, right? Like that. This RBC acts like a plate, that's a disc shape, and receives the oxygen and it turns into oxyhemoglobin, helps in transportation. But what happened due to some mutation, it changed into sickle. And this sickle, unable to receive the oxygen molecule. What I'm drawing in blue color is nothing but oxygen molecule. Unable to receive and carry. So it leads into oxygen deficiency. Okay, and the person will become anemic. It's a flow chart. And here you can see about the hemoglobin genotypes, how it has to be represented. So HbA, HbA. Okay, suppose if you want, it is autosome or allosome it is. It is an autosomal mutant allele. That is why I'm not using XX chromosome like this. Okay, in the previous case, I used like this, XCHXH. It represents the affected person, female with hemophilia. Why I'm not using here XX or XY? Because here it is a autosomal disorder, whereas that is a sex chromosomal disorder, right? So in autosome, directly you can write the part, okay, hemoglobin part gets affected here. That's why HB, HbA, okay? So when I write HbA, HbA, this is a normal hemoglobin. This is the genotype. And HbS is a mutant. So when I write in a homozygous condition, the person is diseased with sickle cell anemia. What will be the heterozygous condition? Let us fill here. HB, yes, actually it should come in superscript, and HbA. Always use the dominant first and then the recessive character. Okay, the affected character. So this is a heterozygous condition. This homozygous suffer from hemolytic anemia. Okay, the medical condition, vascular obstruction or clogged blood vessels. Patient dies due to damaged heart, kidney, spleen and brain, right? Next disorder, agammaglobulinemia. Just read with me. It is a gamma globulinemia. Globin, okay, globin is a protein structure. Globulinemia, a gamma globulinemia. It's a recessive gene disease. Okay, the R globulin synthesis fails here. Okay, that is why we call it as globulinemia defect, reduction. And the symptoms of this are the plasma cell will be deficient or sometimes total absent also. And then a lymph node, unusual lymph node, which leads into a pathway for more infection. So in this disease, AB antibody synthesis will be reduced or fail, and more prone to bacterial and viral infection because the plasma cell is getting, uh, was affected. The plasma cell got affected here, right? And mostly it affect boys. A gamma globulinemia. Next, SID, severe combined immunodeficiency syndrome. It is extremely rare inherited disease affected children. So where this gene get affected? In chromosome number 20. This is important 1 mark question, in which place it is. And what is the gene called as ADA, that enzyme? Adenine deaminase. It affects that, right? The affected children lack immune deficiency. Not lack. Actually, the immune will be deficient here. Okay, which leads into rapid death of WBC. Okay, so you can see here normal immune system and the SID immune system. The normal immune system can fight the infection with the T cells and B cells. Here the T cells and B cells got reduced and they cannot fight with the cells. T cells and B cells, why we are naming it so? Because here thymus, in thymus they get the immunocompetence, whereas B cells in bone marrow itself they get the immunocompetence. So these two are all lymphocytes. T lymphocytes, B lymphocytes. Okay. Next, Huntington's chorea. It's a fatal disease. Again, it is a autosomal, but it is a dominant gene. It's another important 1 mark question. Name an autosomal dominant disorder. The gene location in 4th chromosome, okay, and that gene will get affected. The onset period is 35 to 40 years. What are the symptoms? And this image is nothing but our nervous tissue. So there is a defect in the nervous tissue. It leads into uncontrolled jerking of the body. Okay, involuntary twitching of voluntary muscles. Where the muscles which we can control will keep shaking. For example, our limbs, forelimb, hind limbs, okay, shivering, okay, shivering, or the jerking movement in the forelimbs and hind limbs. That's the twitching of the voluntary muscles, right? Leads to progressive degeneration of central nervous system with gradual mental and physical deterioration. It's actually incurable, okay? Sometimes leads into a thing called depression, hallucination, and delusion, etc. Right? Albinism. It's inherited disorder. Melanin metabolism get affected here. Actually, you can see the picture here. The person, a Negro woman carrying a fat child. Actually, it is because of a disorder. Okay, the enzyme has to produce called tyrosine. This recessive gene, the person who got this albinism will be a recessive. They cannot produce tyrosine enzyme. Because of that, they cannot produce melanocytes, which is responsible for the darker skin. Okay. So let us see here. What are the clinical characteristics? The milky white colored skin. This is number one. And then marked photophobia, inborn error due to mutation. And you can see here absence of melanin in skin, hair, and eyes. Okay. And I'll show you the previous slide. This tyrosine, which converts DOPA into melanin in melanocytes, this function will be reduced or it will not be seen at all. In that case, the person will turn into albino. Okay, sometimes it is like a generalized and localized albino. You can see person with fair skin, okay, even eyebrows, eyelashes, even the hair, everything. You can see your eyebrow, eyelashes, hair, everything turns into milky white color. Okay, some person will have only in the hair, only hair alone will turn into milky white, remaining the skin will have a patched color of white and dark color. So that you call it as localized. Sometimes only in the eye. Okay, you can see here partial albinism, skin and hair, generalized all over the body. Color blindness. Here the person unable to differentiate the color red or green, or sometimes both the color they are unable to differentiate because they have a defect in the cone cells. Where are the cone cells? They are in the retina. In retina we have rod cells and cone cells. Cone cells are responsible for color. Okay, so in that the red cone and the green cone got affected. So they are unable to differentiate between the red and green color, sometimes both the color. It is again a sex-linked recessive disorder. Make a note. Our foremost thing always we have to check whether it is a sex-linked or autosomal linked. And next we have to check whether it is a recessive or it is a dominant. Okay, so mostly we are dealing with recessive, only one are dominant, that is Huntington's chorea. Again, it is a X-linked inheritance. So naturally here female will be the carrier. The last topic is pedigree analysis. Here it's nothing but the family tree to trace whether the forefather have that particular disease or not, like that, right? The study of inheritance of genetic traits in several generations of a human family in the form of family tree, that you call it as pedigree analysis in the form of human family tree. Okay, so from this we can trace out who is having the disease, who was having the disease, who will be having the disease in future. This is a very important topic. Later I'll show you a video about the various problem solving thing in pedigree analysis. Here just we'll see the introduction alone. What is pedigree analysis and what are the symbols denote the characters, right? So here you can see the various symbols with a character. Okay, this represent male and the circle represent female. If it's a single line between the male and female, it's a mating. And then here if you produce a two thing, and actually the circle will be the female and this one square will be the male. These are the progeny. And here if you draw like this, okay, that is a dizygotic twin. And if you connect it, it is a monozygotic twin, identical twins we call it as. And we will number it actually like fourth children or fourth child or fifth child, like that, right? The other one also just go through. Let us see the advantages. It helps in genetic counseling to avoid disorders. There are sayings like when the person gets married with a close relative, there are chances of some genetic disorders. So in that case, they can go and get a help of this pedigree analysis. It shows the origin of a trait and a flow of a trait in a family. Okay, origin of the trait, we told already, we discussed about Queen Victoria. She was a carrier, and then we saw the pedigree chart of a Queen Victoria. From that we can trace who were all the carrier, who was infected, like that. It is important to know the possibility of expressive recessive allele that can cause genotypic disorder. Okay, from here we'll come to know whether the dominant character caused the disease or the recessive. Mostly we are dealing with the recessive gene only here. It predicts the harmful effect of the marriage between the close relative. It is extensively used in medical research. Okay, so we have come to the end of this lesson. Next class, we'll see a new lesson called Molecular Basis of Inheritance. Thank you.